relationship between Parkinson's disease and smoking has been known since 2001, when an article in the medical journal Epidemiology first reported cigarette smokers were 60% less likely to develop the disease, and that the protective effect continued for 25 years after the person stopped smoking. Israeli scientists studied the gene cluster known as CHRNB5, 4, and 3. It appears that as these genes become more dependent on nicotine, they also become more responsible for holding back the progress of Parkinson's. People who smoked for more than 40 years were 46% less likely to develop Parkinson's disease. People who smoked from 30 to 39 years reduced their risk by 35%, and people who smoked from 1 to 9 years reduced their risk by 8%. The primary objective of the study is to demonstrate that transdermal nicotine treatment retards disease progression as measured by change in total unified Parkinson's disease rating scale between baseline and after 52 weeks of study treatment, plus 2 more months washout. Tomatoes, potatoes, and peppers are in the same botanical family as tobacco, Solanaceae. Dietary nicotine or other constituents of tobacco and peppers may reduce Parkinson's risk. Researchers studied 8,000 American men over a 30-year period and found men who drank 3 or more cups of coffee a day were 5 times less likely to develop Parkinson's. In a study with women in Parkinson's disease, coffee consumption was found to have an inverse relationship to Parkinson's, but only for women who were not on hormone replacement therapy. Women on hormone replacement therapy who drank 6 or more cups of coffee a day significantly increased their Parkinson's risk. Researchers fed mice a diet with EHT for 4 weeks. EHT is a component of coffee, and EHT exhibited a dose-dependent preservation of nigral dopaminergic neurons following MPTP challenge. Mice given standard feed were not protected from MPTP. Trigonelline, N-methylnicotinic acid, was named after the leguminous plant Trigonellophenum graecum, fenugreek, from which the compound was first isolated and characterized. Trigonelline in coffee beans is demethylated to nicotinic acid during roasting, and moderate coffee consumption may meet a significant portion of niacin requirements. The trigonelline content was much lower, and the nicotinic acid content was much higher in the heavily roasted coffee bean extract, the so-called iced coffee in Japan, than in the lightly roasted coffee bean extract, the American blend. When fed to rats, dogs, or man, nicotinic acid and nicotinamide are excreted largely as trigonelline. "50 patients with Parkinson's disease stabilized on L-DOPA therapy receive fenugreek capsules twice daily, with matching placebo for 6 months. Total United Parkinson's Disease Rating Scale scores in fenugreek treatment showed slower rise as opposed to steep rise shown by placebo. In conclusion, fenugreek can be useful adjuvant treatment with L-DOPA in management of Parkinson's disease. Discovered in 1932, chlorogenic acids represent a large family of esterified compounds present in green and roasted coffee. During roasting, chlorogenic acids slowly decompose to form caffeic and quinic acid, with about 50% of the original chlorogenic acids being destroyed in a medium roast. Generally, the degree of antioxidant activity of the brewed coffee was inversely proportional to the total chlorogenic acid concentration. The results of the present study suggest that roasting degrades chlorogenic acids to form potent antioxidants, and thus plays an important role in the preparation of high antioxidant, low acid coffee. Four-cafoyl 1,5-quinine, also known as 4-CQL, in roasted coffee inhibits naloxone binding and reverses antinociceptive effects of morphine in mice. Like naltrexone and naloxone, 4-CQL in regular, decaf, and instant coffees temporarily occupies the mu-opioid receptor. The beverage yerba mate is the national drink of Argentina and Uruguay. Its leaves contain 24 vitamins and minerals, 15 amino acids, and abundant antioxidants. In a study of yerba mate and Parkinson's, researchers found an inverse association between intake of yerba mate and Parkinson's, and a potential inverse association is suggested between yerba mate and risk of Parkinson's. These results leads us to hypothesize that yerba mate may have a potential protecting role in developing Parkinson's. In the rat model of Parkinson's disease, green tea polyphenol treatment protected dopaminergic neurons from neurotoxin-induced oxidation. The protection was dose-dependent, meaning the higher the green tea polyphenol dosage, the greater the protective effect. Green tea polyphenols preserved the free radical scavenging capability of both the midbrain and striatum. Brain iron concentrations are known to be elevated in Parkinson's disease patients, and green tea is an iron chelator and free radical scavenger that could be relevant for management of iron overload and oxidative stress. Quercetin is a component of green tea. In an article entitled, "Quercetin Potentiates L-Dopa Reversal of Drug-Induced Catalepsy in Rats, Possible COMT and MAO Inhibition," quercetin, through its COMT and MAO enzyme inhibiting properties, might potentiate the anticatatonic effect of L-Dopa plus carbidopa treatment. This study suggests that quercetin has COMT inhibitory action like entacapone, and MAO inhibitory action like razogeline. Quercetin has poor bioavailability, and bromelain, an enzyme derived from pineapples, helps your body absorb a higher percentage of the quercetin you consume. In an article entitled, "Green Tea Polyphenols, Novel Irreversible Inhibitors of Dopa Decarboxylase," the green tea gallocatechins, EGCG and EGC, were found to be inhibitors of dopa decarboxylase. EGCG acts in a manner similar to carbidopa. Factors which improve EGCG bioavailability include: EGCG has to be of at least 94% purity. EGCG has to be consumed with 1,000 mg of omega-3 derived from salmon and 200 mg of ascorbic acid. This combination has to be consumed on an empty stomach with no caffeine, magnesium, minerals, or milk. One oral dose in the morning after an overnight fasting period, at least 30 minutes before breakfast, and a second dose in the afternoon with fasting period of at least 4 hours, at least 30 minutes before dinner, seemed to be necessary to reach high plasma levels of EGCG. Singapore researchers interviewed over 63,000 Chinese men and women and found there is an inverse relationship between black tea and Parkinson's. The researchers found that by drinking 6 cups of black tea a week, basically one a day, reduced the risk of acquiring Parkinson's by 71%. The researchers concluded ingredients in black tea other than caffeine were responsible for the inverse relationship. No such relationship existed with green tea. Black tea extract has both neuroprotective and neurorescue properties, suggesting that daily intake of black tea extract may slow down Parkinson's progression, as well as delay the onset of neurodegenerative processes in Parkinson's. Dietary polyphenols from berries of similar polarity and structure compared to the polyphenols studied here, theaflavins and theorubigens, are able to cross the blood-brain barrier. L-theanine is an amino acid present in both black and green tea. Theanine calms the brain by releasing alpha waves and GABA, thus counteracting the stress-inducing effects of caffeine. The combination of L-theanine and caffeine improves cognitive performance and increases alertness. The present results suggest that 97 milligrams of L-theanine in combination with 40 milligrams of caffeine helps to focus attention during a demanding cognitive task. Animal neurochemistry studies suggest that L-theanine increases brain serotonin, dopamine, GABA levels, and has affinities for NMDA receptors, in addition to being neuroprotective. Anxiety is a common symptom of Parkinson's disease. In a random double-blind clinical trial, the amino acid theanine outperformed the anti-anxiety medication Xanax. Each of the 16 healthy volunteers took either 1 milligram of aprazolam, 200 milligrams of theanine, or placebo on separate occasions. All participants were tested with all 3 treatments. Theanine but not aprazolam or placebo induced relaxing effects that were evident at the initial measurement of whether a person felt tranquil versus troubled. Epigallocatechin gallate, known as EGCG, is a potent antioxidant in green tea and is present in low concentrations in black tea. The flavonoids camphorol, myricetin, and quercetin can constitute 30% of green tea's dry weight and only exist in trace amounts in black tea. Rutin is a plant pigment that is found in both green and black teas. After the fermentation process, the content of rutin is lowered. EGCG, camphorol, myricetin, quercetin, chlorogenic acid, theanine, luteolin, apigenin, rutin, caffeine are all neuroprotective in Parkinson's disease models and are components of green tea. In an article entitled "Quercetin, Not Caffeine, is a Major Neuroprotective Component in Coffee," researchers tested 100 milligrams of coffee beans and found 280 milligrams of chlorogenic acid, 200 milligrams of quercetin, 60 milligrams of flavones, and 40 milligrams of caffeine. Flavones and chlorogenic acid also proved more effective than caffeine, though quercetin was the clear winner, having the most significant effect. Taken together, the data strongly suggests that quercetin is the major neuroprotective and anti-inflammatory component in coffee, and not caffeine. In a study which compared the standard Parkinson's disease MAO-B inhibitors, razogeline and selegiline, to herbal MAO-B inhibitors, the authors concluded, out of all compounds screened for MAO-B inhibition, the lead was curcumin, followed by chlorogenic acid, and quercetin ranked third. The antioxidant activity of tea infusions was in the order of green tea, white tea, green tea with additives, black and pu'er tea, semi-fermented tea. Green tea becomes black tea when its leaves are oxidized through fermentation. Once oxidized, the main catechin in green tea, EGCG, becomes the polyphenols of black tea, theaflavin, thearubigen, and theobromine. During black tea processing, the tea shoots are macerated to initiate fermentation, in which the enzyme polyphenol oxidase catalyzes oxidation of catechins into quinones by molecular oxygen. Polyphenol oxidase has also been shown to have important applications, such as the use in the synthesis of valuable added products like L-DOPA for the treatment of Parkinson's disease. Catechol acts as a suicide substrate for tyrosinase, causing irreversible inactivation during catechol oxidation. Tyrosinase exacerbates dopamine toxicity, but is not genetically associated with Parkinson's disease. There is a tyrosinase-expressing neuronal cell line as an in vitro model of Parkinson's disease. We have developed neuronal cell lines in which the expression of human tyrosinase was inducible. Overexpression of tyrosinase resulted in increased intracellular dopamine content in association with the formation of melanin pigments in neuronal somata, which eventually causes apoptotic cell death. 10 kinds of Korean traditional teas were screened for their tyrosinase inhibitory activity. Green tea was the strongest inhibitor, and the major active constituents in the tea are EGC, GCG, and EGCG. All catechins with gallic acid group as an active site. The inhibition of tyrosinase by quercetin is a slow and reversible reaction with residual enzyme activity. Among the extracts, black tea had the most potent inhibitory effect, even superior to EGCG. Camellia sinensis water extracts inhibited melanin accumulation and melanin synthesis, along with tyrosinase activity in a concentration-dependent manner. These inhibitory effects were superior to those of arbutin, a well-known depigmenting agent. In an evaluation of the antioxidant and anti-tyrosinase activity of green and roasted coffee extracts, the researchers concluded all extracts also possessed a considerable anti-tyrosinase activity, but less potent than kojic acid and arbutin. Potent in vitro inhibitors of tyrosinase, metal binding fractions of tobacco smoke condensate. Black tea extract has greater inhibitory action than green tea extract, which has greater inhibitory action than arbutin, which has greater inhibitory action than coffee. People with Parkinson's disease are about 4 times more likely to develop melanoma skin cancer, and conversely, people with melanoma have a 4-fold higher risk of getting Parkinson's. People with red hair are more vulnerable to Parkinson's disease than dark-haired people, and people with a history of melanoma are more likely to develop Parkinson's disease than people with no history. Scientists in Boston have provided evidence that the genetic mutation which causes red hair and increases the risk of melanoma might also make the brain more vulnerable to Parkinson's disease. Red hair, which occurs naturally in 1 to 2% of the general population, results from one of several genetic variations. Approximately 80% of people with red hair have a variation in a gene called melanocortin 1 receptor, or MC1R. EGCG inhibits UVA-induced melanogenesis via suppression of tyrosinase activity. These results imply that oolong tea might be effective in whitening and that its inhibitory effect on melanogenesis was involved in the decrease of intracellular tyrosinase at the messenger RNA level. Our results indicate that the inhibitory effect of theaflavin-3 on melanogenesis is due to suppression of tyrosinase protein and messenger RNA levels. We analyzed the effects of chlorogenic acid on cell proliferation, melanin content, and tyrosinase of B16 melanoma cells by inhibiting tyrosinase activity. Chlorogenic acid is a likely substrate of melanin, but the metabolic products of chlorogenic acid may suppress melanogenesis in B16 melanoma cells by inhibiting tyrosinase activity. Consumption of caffeinated coffee was inversely associated with melanoma risk among men, but not women. There were no statistically significant associations between consumption of decaffeinated coffee or tea and the risk of melanoma among both men and women. Green tea extract eradicates skin cancer with no side effects. Short-chain fatty acids and gut microbiota differ between patients with Parkinson's disease and age-matched controls. Fecal short-chain fatty acid concentrations were significantly reduced in Parkinson's disease patients compared to controls. The bacterial phylum Bacteroidetes and bacterial family Prevotalaceae were reduced. Enterobacteriaceae were more abundant in fecal samples from Parkinson's disease patients compared to matched controls. Decaffeinated green and black tea polyphenols decrease weight gain and alter microbiome populations and function in diet-induced obese mice. Green and black tea polyphenol diets resulted in a decrease of serum firmicutase and increase in Bacteroidetes. Black tea polyphenol increased the relative proportion of pseudobutyrovibrio and intestinal formation of short-chain fatty acids. These results suggest that both green and black teas are prebiotics, substances which induce the growth of good microorganisms that contribute to a person's well-being. The new findings show that black tea polyphenols, which are too large to be absorbed in the small intestine, stimulate the growth of gut bacterium and the formulation of short-chain fatty acids. Pseudobutyrovibrio is a gram-negative anaerobic and non-spore-forming bacterial genus from the family Lachnospiraceae. Members of this family protect against colon cancer in humans by producing butyric acid. Cigarette smoke caused an increase in Lachnospiraceae in the colon, however it remained the same in the ileum, the last part of the small intestine. Galactomannan is a carbohydrate-soluble fiber fraction representing 45 to 60% of the fenugreek seed. Bacillus coagulans is a lactic acid-forming bacterial species used as a probiotic. Galactomannan was fermented and utilized by Bacillus coagulans, and significant amount of short-chain fatty acids production was also observed. Furthermore, Bacillus coagulans inhibited the E. coli growth when co-cultured with galactomannan, suggesting competitive fermentation of probiotic bacteria. In conclusion, the mice that consumed coffee had far less E. coli and Clostridium species and far more Bifidobacterium species in their colons than did the mice in the control group. Butyric acid is used as an animal feed supplement due to the ability to reduce pathogenic bacteria colonization. Butyric acid is an important member of the fatty acid subgroup called short-chain fatty acids. Sodium butyrate is the sodium salt of butyric acid. Sodium butyrate improves locomotor impairment and early mortality in a rotenone-induced Drosophila model of Parkinson's disease. Sodium butyrate rescues dopaminergic cells from alpha-synuclein-induced transcriptional deregulation and DNA damage. Chronic constipation is common among people who suffer from Parkinson's, and it has been suggested that constipation may precede the appearance of classic movement symptoms of the disease in some people. In a study about butyric acid in functional constipation, researchers observed a statistically significant reduction in pain during defecation in patients who received microencapsulated sodium butyrate at a dose of 300 mg per day for 12 weeks compared to those who received placebo. Sodium phenylbutyrate is a sodium salt of an aromatic fatty acid made up of an aromatic ring and butyric acid. Neuroprotective effects of phenylbutyrate against MPTP neurotoxicity. Phenylbutyrate upregulates the DJ1 protein and protects neurons in cell culture and in animal models of Parkinson's disease. Sodium phenylbutyrate controls neuroinflammatory and antioxidant activities and protects dopaminergic neurons in mouse models of Parkinson's disease. We expect that phenylbutyrate triglyceride will promote removal of abnormal proteins from the blood into the bloodstream of some but not all Parkinson's patients. We found that the drug increased the level of alpha-synuclein in the bloodstream of all 40 people. The next step will be to give phenylbutyrate for a 1-year period, at least 1 year, to people with Parkinson's. Theaflavins are antioxidant polyphenols that account for 6% of the dry weight of solids in brewed black tea. These results demonstrate that the theaflavin present in black tea possess at least the same antioxidant potency as catechins present in green tea. Black tea theaflavins inhibit formation of toxic amyloid beta and alpha-synuclein fibrils. When compared to EGCG, theaflavin 3 was less susceptible to air oxidation and had an increased efficacy under oxidizing conditions. Thiorubigens are the most abundant group of phenolic pigments found in black tea, accounting for an estimated 60% of the solids in a typical black tea infusion. The bacteria Clostridium botulinum produces the neurotoxin botulinum, the most acutely lethal toxin known, and causes food poisoning known as botulism. Botulinum is so toxic that 50 grams or 1.76 ounces, if it were evenly distributed, would kill every person on the planet. The aim of the present study was to elucidate the mechanism of the protective effect of the black tea extract, the thiorubigen fraction, against the neuromuscular blocking action of botulinum neurotoxin types A, B, and E. These findings indicate that thiorubigen fraction protects against the neuromuscular blocking action of botulinum neurotoxin types A, B, and E by binding with the toxins. As a safe and easy-to-use neutralizing agent for toxins of Clostridium microorganisms, a neutralizing agent comprising thiorubigen as its effective component is provided. "50 years ago, the term thiorubigens was first introduced, and up to now, the chemical nature of the thiorubigens remains largely unresolved, if not mysterious, despite many efforts clarifying their structure. PubChem consists of 3 dynamically growing primary databases. As of November 1, 2017, there are 330,250,000 entries. Pu'er tea is produced by different, more complex, and often lengthy fermentation and heat processes, resulting in a tea with a different flavor and some differences in component ingredients, as compared to the other teas. This rust-brown colored pigment is also found in black tea, but at lower concentrations, 4 to 9% in black tea, as compared to 10 to 14% in pu'er tea. Endoplasmic reticulum stress and mitochondrial cell death pathways mediate A53 and mutant alpha-synuclein-induced toxicity. Hydrogen sulfide is produced as part of the cell's response to endoplasmic reticulum stress. The endoplasmic reticulum is a cellular organelle that is critical for protein folding and, under various conditions called ER stress, protein folding in the endoplasmic reticulum is impaired, leading to the accumulation of misfolded proteins. In ER stress, hydrogen sulfide is released and induces modification to an enzyme called PTP1B, thus preventing the inactivation of another enzyme called PERK, which is a sensor of the presence of unfolded proteins and a critical regulator of the cell's response to ER stress. When the cell is placed under stress, specifically when newly formed proteins are being manufactured in the ER so rapidly that they do not fold properly, rendering them non-functional, The cell must make a decision either to slow protein production to match its physiological requirements, which could restore proper protein folding, or, if that is not sufficient, to commit a form of suicide called apoptosis. Theobromine formulations in the manufacture of proteins for a tyrosine phosphatase inhibiting drug. The present invention uses theobromine or black tea as raw material, and significantly suppressed in vitro activity of protein, tyrosine phosphatase 1b, PTP1b. Pu'er tea extract induces the degradation of Phet family proteins involved in the pathogenesis of amyotrophic lateral sclerosis.